Welcome to Lecture 33. As you can tell from the title, we'll be taking a look at systemic physiology and how it is affected by the adaptations within muscle. Then we'll look at detraining following endurance training, and finally we'll take a look at the adaptations to anaerobic training or sprint training. As in the previous video, I'll start with the objectives for this lecture, and so your pages are 310 to 315. And what we want to do is explain the research showing that muscle adaptations have a strong effect on the respiratory and cardiovascular responses to exercise. That'll be in the first figure we'll take a look at. Then when we get to the detraining effects, we need to understand how that affects VO2 max and the fiber type shifts. And then finally, with anaerobic training, how do the muscle fibers respond? So let's start by taking a look at figure 1311. And this is a series of 5 graphs that show what we would call the systemic responses, or the responses of, say, in this case, the endocrine system, the cardiovascular system, and the respiratory system, to two different types of training that was done. In the first set, well, it was one type of training, but only one leg at a time. So the subjects exercised one leg for 13 days in a row, so 13 endurance exercise bouts, on which measurements were taken, and we can see here up at the top, on day 1, 3, 5, 9, and 13. Then there was a one-day rest period, and then the other leg was trained for 5 sessions, and measurements were taken on days 15 and 17 and 19. And so then what did they measure? Well, that's shown on the y-axis here: norepinephrine, epinephrine, lactate, ventilation, and heart rate. The point of this study was to try to figure out how this adaptation occurs. So we can see that on the first 13 days, we get the decrease in norepinephrine, the decrease in epinephrine over time, the decrease in lactate, the decrease in ventilation, and the decrease in heart rate. And so the question is, why does that occur? Right, I should say, actually, how does that occur? What's the mechanism? Because our possibilities are, well, it's the effects of the training that is taking place in the muscle, those metabolic effects, increased capillarity, increased mitochondria. Or is it a larger effect so that the central command now is being reduced and that we're seeing adaptations in the cardiovascular system, in ventilation, and in endocrine responses? The design of this study, of course, is brilliant because we can study these two systems separately. We can see the training effect in all of these systems from these 5 graphs. And we know that training has occurred in the exercised leg. But what's great about it is now we still have a leg that's not been exercised. And so now we can see the changes in the signaling that will occur if we only exercise the non-trained leg. And so that's what they did. And you can see in each case the arrow that goes up and to the right. So for norepinephrine, it's this, epinephrine here, lactate, it's there, ventilation there, and heart rate there. We can see that on day 15, when now we exercise the non-trained leg, all of those responses go back up almost to what it was at the beginning with the original non-trained leg. Now, the book mentions there is no transfer effect, but I think if you look closely, you can see that in the untrained leg, the second time on day 15, those responses, for most of them at least, are still a little bit lower. At least norepinephrine and epinephrine and ventilation. Looks like heart rate and lactate may be about the same. At any rate, what we see is that now that we are exercising the non-trained leg, all of these responses went back to what looks like to be a non-trained individual, even though we know they just went through 13 days of training. So they don't seem to show any of those responses, which tells us that the changes in norepinephrine and epinephrine and lactate and ventilation and heart rate were being influenced to a great degree by the trained muscle. So it's the effects of the muscle feeding back to the brain and the cardiovascular control centers that are causing this decrease across the board. Another interesting finding in these data is that you can tell, despite the conclusion that there is a lack of transfer of training effect, actually there might be, because if you look at the 5 sessions sessions of the second leg that was trained in norepinephrine and epinephrine, lactate, ventilation, heart rate, all of those decreases, they occurred in 5 days, whereas it took 13 days on the original first leg that was trained. And so it looks like the adaptation is a little faster. So there probably is some crossover. So the bottom line here is that these data show us that endurance training that results in the adaptations within the muscle, it is those adaptations that then are dictating a large portion of what we would call the systemic response, or the whole body response, that is ventilation, heart rate, and the endocrine response, these changes in hormones. So now if we jump ahead to figures 13.12 and 13.13, what we have are the explanations of these mechanisms. That is the peripheral control of heart rate, ventilation, blood flow, and then on figure 13.13 is the changes in central command that might affect the overall responses. So we'll start with figure 13.12, and what we're showing is here the figure essentially begins at the bottom left, where we have these group 3 and group 4 fibers. We've talked about those before. These are these non-myelinated nerves that will carry information back to the spinal cord and then eventually up to the cardiorespiratory control centers. And so in the non-trained muscle, of course, you get lots of group 3 and group 4 activation. So these mechanoreceptors and chemoreceptors, because you're going to get a large change in pH and lactate, things that are going to activate these receptors, because the muscle, this is a large challenge, homeostatic challenge for the muscle. And so in the non-trained state, that you'll really have you'll activate the plus side of this. That is, you'll have a large increase in the traffic going to the spinal cord and then on to the cardiorespiratory control centers. And so what is the result? You get a large increase in heart rate and ventilation, and concurrently you get the decrease here in liver and kidney blood flow. To explain the adaptation effect, we can just assume then that in the trained muscle, again, we're going to have a higher capillarity, more mitochondria. We're going to rely on fats rather than carbohydrates, and so lactate will be lower. The change in pH will be less. So group 3 and group 4 afferents won't be as active, and so we can see then we would have a reduction compared to the non-trained muscle to the spinal cord, and then the traffic to the cardiorespiratory control centers, of course, is going to be less, and therefore your heart rate and ventilation, the increase is going to be less with an increasing work rate. And then you probably also see a smaller decrease in liver and kidney blood flow, just because it's a relatively easier workload. So that explains the peripheral control, the feedback into the cardiorespiratory control centers. If we jump ahead and look at figure 13.13, this shows the effect of central command and the central control of these responses and how they might be affected by training. Going back to the chapters on the cardiovascular and respiratory systems, you'll remember that the central command theory of the control of heart rate and cardiac output and breathing is that the higher brain centers like the cortex, the cerebellum, basal ganglia, these will be involved in the motor programming, by which we will increase our activation of our muscle fibers, our motor unit recruitment, which then, of course, allows us to complete the work by producing enough power, and in doing so, of course, we will increase our VO2. The other side of the central command theory says that there is also then a parallel activation going to the cardiorespiratory control centers that is in parallel with the motor unit recruitment, so that heart rate and ventilation also will show an increase that is proportional to the recruitment of motor units, and then the same with your liver and kidney blood flow will decrease proportionately. So with exercise training, we might also see a reduction in the motor unit recruitment. So instead of having all of these pluses, instead of 5 pluses, now maybe there's only 2 or 3. And that is because if you have trained muscle fibers, with a greater oxidative capacity, you can recruit fewer fibers to accomplish the same amount of power. And then the effect, of course, would be that your input that is parallel input to the cardiorespiratory centers is going to be less, and you might have less ventilation, and then again, a less reduction in liver and kidney blood flow. So those figures 13.12, 13.13 are trying to show the pathways or the mechanisms by which we get this decrease in heart rate and ventilation, and the inputs, and it's a result of lesser input from the trained muscles, and also a reduced drive from the central command. So basically an explanation of the training effects on these systems that occur due to having now trained muscles and with their appropriate adaptations. So that covers the connection between the peripheral adaptations within the muscle and then our overall exercise responses. And so now let's go ahead and take a look at the next section in the text, which is the effects of detraining. Now, if we are highly trained and now we stop training, what are the adaptations that occur? And if we go back to the very beginning of this chapter, we know now this is going to be the application of the reversibility principle. If you stop training, any of the gains you made will be reversed. We'll first take a look at figure 13.14, and this shows us the time course of the changes in several cardiovascular variables that affect VO2 max over a long period of detraining. So we see on our x-axis we have days of detraining from 0 to 84, and then back up to, on the y-axis is our percent change. So at 0%, that's where the baseline would be when we stopped training. And of course, any increase would be in percentage would be an increase, and the decrease in percentage would be a reduction. So let's first take a look at the VO2 max response. This is the green line, and we can see that by 12 days, we have a decrease of about 7%, and that's shown here. And so about a 7% decrease that then proceeds or progressively gets lower to about almost a 20% decrease by 84 days. Now let's look at the cardiovascular variables that seem to be causing this. First off, you see the biggest change immediately is a 10 or even 11% reduction in stroke volume after 12 days. And then after that, not much change at all. You can see it's a fairly slow decrease of only just a couple percent after 12 days. We also see, of course, now the corresponding reduction. This would be the red line in cardiac output. So about a 7 or 8% reduction in cardiac output after 12 days, but then also that remains fairly constant too. Now we remember, of course, cardiac output is going to be the product of stroke volume and heart rate. So if you take a look at heart rate, it actually goes up a few percent. And up to 3 or 4% by 12 days, and maybe even up to 5% increase after 6 or 8 weeks. Finally, let's take a look at the AVO2 difference. After the first 12 to 21 days, we see either no change or maybe a 2% slight increase in AVO2 difference, but then over the next 70 days, this nice but fairly slow decrease down, so you get about a 3 or 4% reduction in AVO2 difference. That's going to be carried out primarily because of a decrease in the number of mitochondria. So let's look at the connection in these patterns. The first 12 to 21 days, notice that VO2 max, which is in the green, decreases 7 or 8% into this area, and that is paralleled almost precisely by the decrease, here's the red line, in cardiac output. And that's primarily being driven by what? The decrease in stroke volume. from the time period of 21 days until 84, we can see the reduction in VO2 max. I'll do this in black right here. This reduction in VO2 max, that green line, is almost exactly parallel with the reduction in AVO2 difference right here. And so we can tell which is driving which. In the first 12 to 21 days, the reduction in VO2 max paralleled the reduction in cardiac output, and then the next 60 days, we can see the reduction in VO2 max matched essentially exactly the reduction in AVO2 difference. Hopefully that reminds you of the time course that we looked at the beginning of this chapter when we looked at cardiovascular adaptations, or the time course for changes in VO2 max with training. Over the first 4 weeks, we saw the 10% increase in cardiac output and the corresponding increase in VO2 max, right? And then over the next 28 weeks, there was the further increase in VO2 max that was driven almost entirely by the increase in AVO2 difference. Well, this is the exact same sort of response that we see here with detraining. First in cardiac output and then later in AVO2 difference. In addition to these changes in cardiovascular variables, we will also see a change in the fiber types. And this will be opposite of the training effect that we saw. Remember, we will see a fast to slow shift with exercise training. And with detraining, we'll see a more of a slow to fast. And so as our book points out, over many weeks of detraining, we saw a decrease in the type 2A fibers, those fast oxidative glycolytic, from 43 to 26%, and then a corresponding increase in type 2X from 5% to 19%. So they're getting less oxidative. Of course, that will also have an effect on the submaximal endurance performance, which of course is going to suffer as well, especially as the AVO2 difference and lack of mitochondria become more profound. Let's take a look now at figure 13.15. This also shows a time course. So on the x-axis, we have weeks of training or detraining, and on the y-axis, we have mitochondrial content. This is actually data from a publication by my graduate school advisor, Ron Turyoung, the author of, one of the authors of the article we just discussed on Monday. If we look at the red line, what is this showing? This is showing your mitochondrial content with training. And it shows that after about 5 weeks, we can see that basically you've reached the asymptote, you've reached the peak mitochondrial content at about 2 times the normal level. And then you can see what happens if you go week 6, 7, 8, 9, and 10. Basically, it levels out, and there's not much of an increase after that. But look what happens if we do the detraining. If we take the purple line, after 1 week, from week 5 to week 6, this is this period marked A right here, we can see we lose about 50% of your mitochondrial adaptation. That then slowly, progressively will go a little further as detraining continues. But if you only take 1 week off and then start retraining again, we can see the green line, the retraining, it takes a full 5 weeks to return back to what you would have been doing had you been training. So we lose the adaptations pretty quickly with detraining, and it takes a while to get them back, unfortunately. So up until now, we've been talking about endurance training and the adaptations in both cardiovascular and muscular systems. But so now let's turn our attention to anaerobic exercise training, or otherwise known as sprint training, or sometimes interval training. This can take on many forms and has lots of variation. So what really we can look at are intervals that are super short and super high intensity, anywhere from 5 to 10 second durations, or even those up to 20 to 30 second durations. What we find is that you'll see an improvement in performance, and then we'll also have some changes in the metabolism of skeletal muscle. So first, if we look at the improvement in performance of something like a sprint test, like the Wingate test you can do on the bicycle, you're going to see after 4 to 10 weeks of training anywhere from a 3 to 25% improvement in performance. And this is quite variable and probably is mostly determined by genetics. as I just previously mentioned, the changes in skeletal muscle that we're going to see are going to be predicted mostly on the type of training that's done. So if you are doing highly sprint training, 5, 10, 15-second bouts, as opposed to 30-second or 1-minute bouts of exercise, so that would be sprint training versus sort of interval training. That's going to dictate the type of responses that we'll see. But we do see 3 main adaptations, and that's an increase in the muscle buffering capacity, a hypertrophy of the fast twitch fibers, and then changes in the metabolism, that is specific enzymes, myokinase, creatine kinase, and the enzymes of glycolysis. So let's take a look at those. First, let's look at improved muscle buffering capacity. This is the ability to handle acids. So we know with high intensity exercise, we're going to be producing lots of lactate and lots of acid. pH is going to drop inside the muscle cell. The adaptations that happen here are we get an increase in what we call intracellular buffers. Those are molecules that will literally bind the protons, H+, the hydrogens, that will then take it out of solution, and that reduces your increase in the H+ concentration and the acidity. We also increase the transporters that transport proton through the sarcolemma and out into the extracellular fluid. Both of those will help keep intracellular pH higher after training, whereas compared to before. And so with both sprint training and high intensity interval training, we get an improved muscle buffering capacity, and so we can handle acid a lot better. We can tolerate it. A second major adaptation within muscle to anaerobic training is the hypertrophy of type 2 fibers. And so this will be specifically like something like strength training. You're doing high force, whether it's revolutions on the bike or running, and your type 2 fibers are being recruited to a large extent, and we will see an increase in their size, both type 2A and type 2X. The main result here is that you'll have an increase in strength, and this probably more than any changes in metabolism will be the reason for improved performance, let's say on whether it's sprinting on the track or on something like the Wingate test. With bigger, stronger fibers, you can produce more power, regardless of what your rate of ATP provision is. A third adaptation with sprint training is that you will see an increase in the enzymes myokinase and creatine kinase, and then if we do something longer, say 30 seconds, 20 to 30 seconds or more, also the glycolytic enzymes will increase in their activity. Finally, if you're doing interval training of 30 seconds or more that's repeated, you will also see an increase in the oxidative capacity of those muscles that were recruited. And so essentially you can have a slight improvement in endurance performance. And the really big improvement here might also be in the recovery of the muscle. The greater the oxidative capacity, the faster you can recover and better perform on repeated sprints, which can be important in those intermittent sports like soccer and basketball. So that takes us to the end of Lecture 33.